It's my privilege to welcome you all again here tonight for the second of our public lectures as part of the Natural Science Section's annual conference. This year's conference, as you probably all know, is entitled Building Bridges from Natural Science to Spiritual Science. Last night we heard on the theme, is there a place for the spirit in physics and chemistry? On the importance of epistemology for natural science. Tonight, Dr. Peter Heusser is going to move us farther forward as we'll start bringing in questions of life. Tonight's theme is there a chemical or genetic explanation of life? A closer look on causality and agency in modern biology. Please help me welcome Dr. Peter Heusser. Thank you.
Thank you, Andrew. Dear friends and colleagues, chemical explanation of life. In 1842, Émile Dubois-Reymond one of the leading physiologists who actually founded, this group of physiologists, founded modern medicine, wrote in a letter to a friend, Brücke and I, Brücke is another one of these physiologists, Brücke and I, we have sworn to ourselves to enforce the truth that no other forces are acting in the organism than the plain physical chemical ones. And in a famous lecture of his in 1872, he said, understanding nature means to attribute changes in the physical world to movements of atoms which are caused by nuclear forces, or to break down the processes of nature into the mechanics of atoms. Now this is, as we know, the paradigm of reductionistic science, which also prevailed in the 20th century. After the detection of DNA as the substance of inheritance and the discovery of the DNA double-strand helix and the genetic code in the middle of the 20th century by Francis Crick and James Watson, molecular biology became the big thing till today. And everything in the organism was and still is explained from, lastly, from chemical and physical principles. So again, the chemical explanation of life, as Francis Crick himself put it, is still the explanation principle also for life processes. Now, we know that this chemical understanding of genetic understanding of life is based on the notion that the structure of the DNA, a chemical substance, determines the structure of proteins, and that proteins are used either to build structures in the organism or to guide functions as enzymes in cells, organs, systems, organ systems, and in the whole of the organism. In short, the DNA is believed to contain the information for functions and forms, actually for everything that happens in the organism, for the overall Bauplan, the blueprint of the organism, which then is handed over to the next generation of organisms. So that's still what is, in essence, being taught in medical schools, for example. Now, if we acknowledge the notion of the genetic code consisting in a sequence of base triplets on the DNA, which is then translated into the sequence of amino acids in the proteins, then we are entitled to ask the question, what exactly is the information contained in the genes, and is it really true that all information of the organism is coded by the genes? Now, a gene is classically understood as that portion of the DNA which determines the primary structure of a protein. That's what we classically understand when we talk of a gene. Now, I would like to ask, who of you is not familiar with the expression of genes, how information gets from the genes to proteins? Who is not familiar with that? Because then I would briefly, okay. So I briefly, very, very simply, oversimplified, I try to explain what happens here. So first of all, we have the DNA, which are long thread-like molecules, which consist of three main chemical components, so to speak: sugars, phosphates, and bases. There are four types of such bases, which have certain names. That doesn't matter now, but these four bases are structured in such a way that always two are complementary to each other, right? So now, if this is to symbolize a thread of the DNA, and this is to symbolize these bases, so essentially there are four kinds, right? Then in the double helix, we have always the polar base on the other side. Okay? So that means that always two belong together. So then they have a certain bonding, and the whole thing, these two threads are then put into a helical form, right? Okay. Now, where is this thing to extinguish? This is right. So, let's just take one. Now, we always have three of such bases which code for, we have a protein here which also is a thread, well, of peptides. Then, thank you. Then we have amino acids, and here we have the so-called base triplets. So three bases are, let's say, are like the equivalent or the correlate to specific amino acids. Okay. Now you can imagine that if we have these different bases, and if we have four kinds, you have different combinations to make triplets, right? So essentially you have more than 20 possibilities, and depending on this, you have a certain sequence of amino acids. Is that clear? Okay. So genetic information means that these triplets determine what amino acids will be on a protein. That is essentially what genetic information is about. So, but this process happens, this translation of this information happens in certain stages, and I will quickly depict how this happens. let's erase this here. And we don't need anymore. Now, if we have a cell, we have a nucleus in the cell where the DNA are contained, and we have the plasma with different organelles. That's where the proteins are produced. So proteins, DNA. Now we have a process, first process called transcription. What happens now is that a so-called messenger RNA is produced. This also consists of nucleotides. There's just one of these bases is different, but essentially it's the same thing. And these also are polar to these bases here. So that means there's a synthesis of the messenger RNA through the activity of certain enzymes, RNA polymerase it's called, and so there comes into being a strand of messenger RNA. They're not so long, and so they have the corresponding polar bases to what we have here, right? So the production of this is called transcription. Transcription. So now, but this happens in two steps actually, because what first happens is that the so-called precursor messenger RNA is produced, and this has different portions, which are equivalent to different portions here. And I go like this and say, this is a portion, this is a portion, and then there are other portions in between, symbolized by different colors. So the corresponding portions exist and also on this RNA. And now happens the following. Some of these portions are excised. Is that the right word? Are excised. I just go like this. And the other ones are spliced together. Okay, so those which are spliced together are called exons. Exons because they have to go exit, right? And the others are called introns. And these introns, they are not used anymore in this process, but they are degraded. And so this is what happens here. And then this messenger RNA travels out into the cytoplasm. And there we have little organelles which are called ribosomes. And here, this messenger RNA is used as a template. It attaches somehow to this ribosome, which are protein structures. And then the following happens. Then we have the so-called transfer RNAs, which are very small RNA, and each of them drags to this organelle an amino acid. So that means, and these amino acids are then attached to this. We have another color. Yes, here we have one. So these are the amino acids, right? So they will be attached to these base triplets of the messenger RNAs per 3 bases, 1 amino acid. So eventually what happens here is a chain of proteins. Okay? Now this is then detached, of course, from this template, and that's then the primary structure of the protein. Okay. Now you see that the sequence of the amino acids on the protein is corresponds actually to what to the DNA, but not to the whole, let's say, as such to the DNA, but only to these structures here, to the so-called exons on this strip. Okay. So then another process happens, and that's the formation of a three-dimensional structure, which is necessary in order to have a functional protein. Okay. So this process is called transcription, and the second process is called translation. And this is the folding up process to a functional protein. Now, if we say a gene is the portion of a nucleic acid which contains, so to speak, the information for a protein, then the question is the information for a protein on the DNA? No. What is on the DNA as far as this so-called information is concerned?
Templates.
Yes, templates, but not what exactly?
The information for
The primary.
information
sequence.
Yeah, primary sequence.
Yes, but if we say that the gene is that nucleic acid which contains the information for the primary structure of the protein, we do not find the primary structure of this protein here, but only mosaic kind of portions of it, right? That's very, very important. So actually, the gene, the actual gene, is not the DNA, but is the messenger RNA. And that's why, for example, Evelyn Fox Keller, a very eminent geneticist, talks of the functional gene or the true gene. The true gene is the finely produced messenger RNA. That's very important to see, okay? So, now, that means it's not true that the information just lies on the genes. And as we will see, another thing is also not true. When we come to the confirmation here, we will have a big problem. We talk about that afterwards. But one thing I would like to say, we tend to think that the DNA is a fixed structure, which is stable. And in this stability can be given over to the next generation. This stability comes only into being through the action of enzymes. We have a constant, for example, a control of the stability of so-called repair mechanisms. Well, it's not mechanisms, but repair processes which are so-called guided by proteins. So this is a constant activity that this happens. Then the winding and unwinding of the double strand helix is guided by enzymes. It's a product of enzyme processes. The production of messenger RNAs is a production of enzymes. Enzymes have to do that, so to speak, or to lead that, to guide that, to regulate that, etc. So we have enzyme processes all over. And the so-called stability of the DNA is due to enzymes. So enzymes do all of that. We have to realize that. I will come to that later. Also, let's say the following is important to know. There are so-called alternative forms of splicing. This means that from the same primary or precursor RNA, different messenger RNAs can be produced. In that, for example, the sequence of these portions is not the same, is alternative. Also, this is guided by proteins. And this is one of the reasons why, out of these about 25,000 genes, so to speak, that the human organism contains, approximately 1 million different proteins can be produced, according to what the scientists have calculated. So there's a multiplicity of proteins, and from this you see that these genes are not just fixed things, but they are produced on demand. They're produced on demand. So there's a constant activity in our organism which always produces true or functional genes, which are then the templates for the primary structure of the proteins, right? So we have to look at the dynamic of this, and it is extremely dynamic. For example, we know today that these DNA structures are constantly changing. We have the doubling, they are, separated, and we have the doubling and all that, and they make all kinds of movements, so that today one talks of dancing DNAs. They're dancing, sort of. And so but they do that by virtue of proteins which regulate. So the result is that genes are an active product of the organism. And also the DNA is, in its stability, etc., a product of the activity, a constant activity of the organism. There's never any rest. And there is nothing which is just stable. That doesn't exist in the organism. Nothing is stable. Everything is exchanged all the time with the help of proteins. Okay, now we come to this problem. Because with this folding up process of the protein to a form which is called the conformation, where we come to those functional, let's say, regions of a protein, for example. Here, this region which might be responsible for a enzymatic activity. They are always specific. This is a high specific, right? So the question is, how does this folding process happen? How it is generated? Now the usual explanation is that this is it's determined, the tertiary, it's also called the tertiary structure. This is the primary, and this is the tertiary structure. It's determined by the primary structure. Why does one say that? Because one knows, for example, that if we have here an amino acid which contains, let's say, sulfur, and here also, then if winded up to a helix conformation, for example, then there will be a what do you call it? Double bond. Disulfide bridge, right? Which eventually stabilizes the helix or the structure of the protein. Because of such side groups here, which have to do with secondary conformation, one thinks or one believes or states that the tertiary structure is determined by the primary structure, or as Löffler-Petrides, one of the very well-known leading biochemist in Germany says, the primary structure, that is the amino acid sequence of a protein, must always contain all the information for producing the correct, unique spatial structure of the protein. This one. That's what they say. So one thinks actually conformation is a bottom-up process. Of course, the conformation is equally let's say, an emergent structure, as comparatively, the snowflake structure is an emergent structure. So there is a new form imposed on the primary strand, which is actually not contained in the primary strand. And why it cannot be contained in the primary structure, we will just see in a moment. But one thinks it's a bottom-up process. So then the question is, where exactly is the information for this three-dimensional structure? There must be an information for the three-dimensional structure. What is the information for a three-dimensional structure? It's the geometry, the geometrical law of this structure, right? That would be the information. But this law you don't find anywhere. Just as you don't find the law of a snowflake when you look at the water before it is formed into the form of a snowflake. This tertiary structure is emergent, and we have seen yesterday what emergence means. It means that you cannot calculate or derive the tertiary structure or the emergent structure from a knowledge of the submergent or structures of the lower level structure. So sulfur-containing amino acids are not the cause of the primary structure, but conditions for the primary structure to become stable. That's all what's to them. So if we now add or remember what we have dealt with yesterday, we would have to say, well, the law of the conformation is a law in its own right. It's an emergent law that enacts itself. So we could talk of active information, but now not only in the sense of quantum physics on the sub-molecular or subatomic level, but active information of the law of conformation. It's an active conformation. It's a form principle. It's a physical active form principle, which enacts itself. This is what you can infer from all that we have treated since yesterday till today. And this would not be a bottom-up process, but a top-down, right? This would necessarily have to be. Now, and this really seems to be the case. And this we can see when we look at the problem of multiple folding options. Multiple folding options. For example, already in the 1993, Fischer, a German biologist, said, due to the large number of possible arrangements of the atoms to each other, the time requirement for the transition of a small protein to its native state, which means this would be the native state, is already astronomically high. When you can also consider that all the theoretically possible positions in the conformation space have to be crossed along the folding path. Nevertheless, some denatured proteins can be refolded to the native structure very quickly, in a matter of seconds. The polypeptide chains can obviously fold along a preferred reaction pathway without having to search the entire conformation space available. A preferred pathway. Well, if it's a bottom-up process, it couldn't be a preferred pathway. Because one has to see that there are multiple, let's say, deviations possible, which are, or branches, you know, you could fold to the left or to the right side with an energetically equivalent, let's say, condition, right? So it's not determined where it goes. So obviously there must be some kind of really preferred or even directed process. Löffler-Petrides, that's two authors which I already mentioned. They, in Germany, medical students have to study from a thick book, biochemistry of Löffler-Petrides, and in there you read the following about the problem of the folding of protein structure. If each possible individual conformation of the polypeptide backbone of a protein of only 100 amino acids was tested for its functionality. So, you know, you could test all these different conformation pathways. This would take at least 10 to the 50th, 10 to the 50th, that's how it says in the text, 10 to the 50th years until the native biologically active form of this protein was reached. This thought paints to the fact that protein folding into the correct spatial structure must be based on a goal-directed process. You see? Goal-directed. It couldn't be goal-directed in a bottom-up process. It would have to go through all these different possibilities, and then the functional ones, the fittest ones, according to a selection principle, would be selected, and then finally we would have about 10 to the 50th years. How old is the universe? Where are the physicists? How old is the universe?
Supposedly 4 billion years.
4 billion. So that would be 10.
13 billion.
Huh?
14 billion.
Earth is 4 billion.
14 billion.
Yeah,
Okay. So billion is 10 to the 9th, right?
Yeah.
So we could several times the whole creation, right? Just to have a secure protein of only 100 amino acids, and that's a very short, that's a very small protein. But we have about 1 million different proteins, right? With all their different structures. Just imagine that. So here, again, we come to a limit of natural sciences where modern investigation possibilities lead to the impossibility that these simple explanations that, you know, there are nothing else is acting in the organism, that the plain physical-chemical processes, that this actually cannot be true. There must be something else, something which is goal-directing. So we could, taking up the theme of last night, we were talking about the causality of Aristotle. Causa formalis, formal cause. But here, in a living system, this causa formalis is at the same time a causa finalis. It's a final cause, which enacts itself to have a goal. And we know from very modern experiments that apparently in large proteins, there can be 2 or 3 different pathways, but all leading to the same conformation. Right? So it is directional, but as in a living system, if a child breaks a bone, then the bone and the child has to be in bed for a long time, then the bone won't grow for a certain time. Then what happens? Instead of the bone growing like this, you know, according to our statistical means of, you know, these percentiles, the bone goes like this. It stops growing a little bit, or it's the growing is retarded. But what happens afterwards? Goes back again. Goal-directed process, even if we have a deviation. Typical for a living system. So what happens here, actually? How can, look at these astronomical figures, how can such thing happen? So, now, in a way, we can say, yes, the formation or the production of the conformation process is similar to a crystal. In that a conformational form principle enacts itself in a comparable way as in a snow crystal or in any other crystal, a form principle enacts, as we saw yesterday. But, and Steiner even says also that if the human organism wouldn't be living, we would be a big crystal, complex big crystal. And it's not only the proteins, it's also the DNA, the lipids, if you take the macromolecules, it's the cells with their structures, membranes, and all that. This would all be in a crystallized form, right? If it wouldn't be living. But it lives. And it lives with help of the proteins. And not only, I mean, it's not what we shouldn't think that the proteins some kind that have to do with life and everything else is dead, and that would be not the point, but the proteins apparently have a special role in that whole process. So it is, in a way, it's like a crystal, and in fact, also a modern biology treats proteins as crystals. Like if you determine the structure of a crystal, you do it with the same technical procedures as you determine the structure of an inorganic crystal with crystallography, X-ray crystallography, right? But more and more, one sees that these are not really crystals. They are living in a certain way. And for example, one of the first proteins which have been determined in their three-dimensional structure is myoglobin. So this is a protein which has to do with muscle, muscle protein, myoglobin. Now, the interesting thing is that now myoglobin it's not a protein that has to do with a movement process, but with the, making available the oxygen to the muscles, right? And these myoglobins, they also have a heme structure, as we learned today. right? And they have to take up the oxygen. And now one has found out that these myoglobins, if you take the tertiary structure that they have, they could not take up the oxygen. So what do? We know today they are flexible. The conformation is flexible. It's like a fluid thing. And when oxygen comes, floops, it changes the conformation so that oxygen can be taken up and can reach the end. Heme, right? For example, this is one thing which is already known for a long time, actually. So this fluidity or this flexibility of proteins is now becoming a big theme, because today we know that actually these conformations are also not stable. They're moving all the time. They're also oscillating in an incredibly great, let's say, speed. So it's in the order of picoseconds. Picoseconds. How much is it? Billions of seconds or something like this, huh? Right? So it's an oscillation. It's a change of a spatial structure in time. It's a change of a spatial structure in time, which is coordinated in such a way that, for example, in this myoglobin, there are several parts of this conformation which have to be coordinated in order for the oxygen to enter the protein. So you see, coordinated in time, in an incredible speed. This cannot be the effect of a purely physical crystalline structure, right? It's impossible. So it must be something else moving here. And also, if you look at the following, for example, what do you see here?
Network.
A network. What do you think it is?
Protein cycle.
Pardon?
A protein cycle.
Not a protein cycle, yeah?
I recognize it.
Yeah, what is it?
Metabolic pathway.
Huh?
Metabolic pathway.
Why do you recognize it?
I read it in the book.
Okay. This is a citrate. I never know how to pronounce it.
Citric acid. Citric acid.
Cycle.
Circle, right?
Yeah.
Citric acid circle.
Cycle. Yeah.
Of course, this is a schematic depiction of what a liver cell does or contains, just in terms of the biochemical pathway. If you take the whole liver cell, it has much more. It has, this production of proteins all the time. Because now we have to think that all these different dots here, these dots represent chemical, let's say, stages of these processes. And between these chemical stages, everywhere we have to imagine a protein acting as an enzyme. This is everything is enzymatically regulated. So now look at this. In order to have such a thing, what a coordination must be at place. How to produce these hundreds, if not thousands, of proteins at the right time and working at the right place. How to do this? How to coordinate? And it's not like a Swiss clock or a Swiss watch, which is also, well, this happens to be a German watch. Actually a good one. So even as a Swiss watch. So, but this is a fixed thing. This is a mechanical device, right? And it is in a solid structure And this is a rather simple structure that we have here. In our computers, we already have much more complicated structures, but they are also, all of them are in a fixed, rigid, solid structure. Now, this is not a solid structure, but this is a liquid structure. We were talking about today, where is Frank? Also about these things, that in life, of course, everything is quasi-liquid. We have about 65% water, and the rest is proteins and all these other things. So now, how to imagine an order of such a complexity in a liquid state? Now, if you would buy this watch in a liquid state, would you be satisfied? You would watch out the shop, and it would already be, everything would not be in place anymore. How is this structure being kept up in a liquid state? You see, we have to think concretely, take the consequences of what modern science has brought, and then think about that. How can this be? And it is really coordinated in such a way that also this is coordinated in space and time with oscillations. I just read a short passage. Oscillation. Thank you. Yeah, my pronunciation is not always optimal. No, it's not. Oscillations have likewise been noted in key signaling pathways, and there is growing evidence for the importance of an oscillator's frequency in controlling downstream biological events. And dynamic interactions between oscillators with different frequencies may be a key component of signaling crosstalk in cells. Oscillators, again, a time structure supersedes a spatial structure. And all these spatial happenings are coordinated in time, in space and in time. This is an important point because here we have an orchestration apparently, which if we would transform it into notes, we would have a musical system. We would have eurythmy. So our organism performs eurythmy, right? So the DNA dances, and this all oscillates, this is an orchestra. Well, but if we have an orchestra and one talks more and more about orchestration or concertation in biology. Now, what does an orchestration necessarily need? Yes, conductor. Who is the conductor? So these are the problems. And now, if we think of that, and that always the proteins are there. The proteins are the ones by whose mediation the DNA have their stability, their expression, and all that. And the proteins themselves are subject to an oscillatory process where they have conformational change. It is even the case that conformations can change to new conformations, and that two different specific reactions can be catalyzed by the proteins. How can this happen if the primary structure is responsible for the tertiary structure? It's not responsible. So here we have a form principle which cannot be derived from the genes, but neither from by DNA, neither from the messenger RNA, because it simply cannot be derived from primary structure. It is something which a law which enacts itself, and we see these are spatial laws. Conformations are three-dimensional spatial laws, but they are subjects himself to a time, a temporal change. And this temporal change must have a lawfulness, because if this is to be coordinated in time, you can imagine what kind of a logistic is necessary in order to do that, right? So, who is the conductor? It's that, I would say, association of temporal laws, because if the temporal structure is again emergent from the spatial structure, then there must be temporal laws which are just as realities as the spatial ones, right? Also in the sense of what we had yesterday. And because these laws are very, very complicated, it's a complexity of laws, but it's really like an orchestra which is organized, so it must be an organization of temporal laws. And temporal laws are by nature different than spatial laws. These are not physical laws. Physical laws, they enact themselves as space, in the form of space. Temporal laws, they act on this space, but they are themselves not spatial in character. They are temporal in character. This organization, whose laws is temporal in character, Steiner used another word, also a specific word, in order to distinguish from the physical types of laws. He talked of etheric laws. So an etheric organization, or as Steiner also says, etheric body, but body now not in a physical sense, but in a sense of a force organization. These are lawful forces, a lawful force organization acting in time. Therefore, the etheric body is a time body. So apparently, if we take this as well, this is our concepts from spiritual science, but you see how much sense it makes to compare that with what natural sciences has found up to date. These findings become more and more interesting, more and more interesting. In fact, most modern science, the finer our also instruments become, the more we can go into the details, the more we can be astounded what kind of a wonder this is. It's like today in Europeans, we really have to wonder what happens here. And we come to our limits with understanding this from our ordinary way of explaining things. It cannot be plain physical, chemical laws which act here, but we also have something which does this temporal coordination. Now, on this basis, I would like to read from Rudolf Steiner, Rita Wegmann, Fundamentals of Therapy, Chapter 9, The Role of Protein in the Human Body. Just some few sentences. Protein is that substance of the living body which can be transformed in the most varied ways by the body's formative forces, so that the forms of the organs and of the whole organism appear as a result of the transformed proteinaceous substance. To be suited for such use, protein must have the ability, the moment it is called upon in the organism, to serve a form demanded by it, to lose whatever form may come from the nature of its material constituents. These formative forces are bound up with the etheric body. Protein is ever on the point of either being taken up into the activity of the etheric body or falling out of it. And then it has to be degraded. So if if we take this and then compare that with the fact that indeed all these operations in life are, we say, regulated by enzymes, but the enzymes don't regulate anything at all. They are instruments to regulate something, and as we see, they are themselves regulated in a temporal form and in the overall process, in the singular cell, but also in the organs, in the organ systems, and in the whole of the organism. So we have to imagine a temporal force organization which is responsible for all that happens in this very complicated process. Now I would like to add now another piece of interesting research, and this concerns morphogenesis of the Drosophila fly, which in principle shows similar things as happen also in the organisms of mammals and in human being. What I would like to show you is something which led to the Nobel Prize in 1993. By this guy Volhard and Wieshaus. This guy Volhard is a German geneticist in Tübingen, Wieshaus, an American geneticist. They have worked together and they found out how the body plan, the basis of the body plan of the Drosophila fly is determined by genes. From what we said, we would have to infer that the body plan actually is that physical structure which is enacted in time this is also, of course, a final process, right? Took embryology. The organism is produced through 9 months with various changes. Everything is living, transforming, etc. But it's a goal-directed process. So, and but then the plan itself is, let's say, there's a physical plan, and there's of course also a time plan, because we know this has a certain time lawfulness that takes so many years or months, etc., for the organism to grow like this. This is all lawfulness. That's temporal lawfulness, right? So and the way we argued till now was that we said, we make finally responsible a set of physical forming principles, which are then which are the essence of the physical body, which are regulated or, let's say, superseded by a set of, or an organism of temporal processes. So this is physical and very body. So formative principles, spatial and temporal formative principles. So, and principles are by nature ideas, right? Laws. So they are spiritual. If we add here what we had yesterday. So actually we refer to a spiritual principle, which is at the basis of the Bau plan of the organism. Now, Nusslein Volhard, they found, well, they didn't think of anything like that, but they said, well, they found the following. As you all know, this is Nusslein Volhard, it's a publication of herself. That organism, this is a larva of this fly, fruit fly, that bothers your oranges at home. right? That the structure is very strict, it's very exact in all insects, right? So the question is, how does the planning, the structure, this bug plan of such an organism arise? And she found that there are varieties where the head region is not developed correctly, and that a certain gene is defect, decoid gene, decoid, or bicoid, I never know how to pronounce that, decoid. Bicoid. And here, then there is like a gigantic head, but no trunk. Again, a certain gene mutation or defect could be detected. None could be identical. So they thought, okay, we have genes which are responsible for the production of the bowel. And now we know that when the egg of a fruit fly is fertilized, this would then be the fertilized egg. About this structure, it has one nucleus, and here the membranes and everything. And this is a stage about which I will talk right afterwards. But then this nucleus divides, and so the nuclei which are produced align along the margin of this egg in such a way that there is still one cell, but with many nuclei, right? This is what is called a syncytium. Is that the right word? Syncytium. Yeah. And then only after this has happened, then there will be cells which will then be divided, and then have the multicellular organism, which eventually will develop the precursors of the later organ system in the gastrula. So that's the way this development comes into place. And now we look at this stage. This stage, that's still in the mother organism of the fruit fly. Now what we know is that a so-called maternal pattern, a maternal pre-pattern of messenger RNA in the front decoy, and at the end none messenger RNA from the mother organism are deposited here at the fringes, or is the right word? Oh yeah, here now, and here, I don't know now which one it is. Messenger RNA. And then what happens is the following. I think this is, I'm not sure now, because it's very much at the circumference. And then what happens that in the child organism, this messenger RNA is translated into a protein, so in a decoid protein, and in the mammals protein. And these are the proteins. And these proteins then will travel through the organism in this and that direction, and that in that direction, by diffusion. And so the hypothesis was that if we have different such substances, proteins, which stem from the maternal pre-pattern, then we would arrive at different, let's say, sections, right? Because these might not travel all, to the same place. And in fact, they worked out, they investigated 4 different such messenger RNAs in their proteins. And they could show that with these 4 genes and their products, the axis from head pole to the tail pole and from the belly to the back is determined by these 4 genes, right? Okay. And her hypothesis was, or these 2 authors, that just by simple diffusion, this would come about by simple diffusion, passive diffusion. For example, you pour a liquid into a cloth or in a piece of paper, laboratory paper, and you let it, and you see how, filter paper, thank you. Then you see how the front of the liquid travels. Why? Because you have a high concentration at one point, and then until this concentration is wears out, and depending on the electric properties, on biochemical or chemical properties, and on the properties of the substance in which these substances travel, you might have so and so great distance, right? Okay. So this would be determined purely by chemical and physical processes, purely. And that was their hypothesis, that this would happen. And they could show, yes, in fact, you do have a diffusion, right? But what you see here, for example, the diffusion is rather diffuse, or let's say this would be natural for a natural diffusion. Here you have a substance which has through the whole organism. Here, so if you combine that, then you get some kind of a pattern, right? So, and they found out later. One found out, yes, indeed, it depends then on the concentration, certain concentrations, and how these cells will be activated, because nanosymbicoid and also the bicoid and the other substances are morphogen substances or morphogenetic. Morphogenic substances, right? So they enter the nucleus here, the nuclei, and then they act as regulators of gene expression. And depending on the respective concentration, different genes will be activated. Because you can imagine very quickly that, of course, every cell here along the line must have a different concentration ratio of these 2 substances, right? And it is known, it was also then the hypothesis that indeed it depends on this ratio how the genetic expression is regulated in these cells. And so then afterwards there are 3 different, let's say, generations of genetic processes which determine the broad regions and then the fine regions, and then finally the appendices to these fine regions like legs and wings and so on. So 3 different generations which in sequence have to be activated, but the initial thing is that the ratio of bicoid and nanos determine how the next step will be. And so they said, okay, we found out that through diffusion, in a sense through a chemical process, this pattern of the organism comes into existence. Chemical explanation of life, the life process. Now, if you hear that, what do you think about this? What do you think? If you hear that supposedly a passive diffusion is responsible by virtue of simple chemical physical processes to determine this exact bow plan of such a fly. No. What would you say? What would you demand? What actually should happen in order for that to become true? How can this exactitude be arrived?
Well, it sounds like it's not in the genes, but it's on an outside entity that organizes.
Yeah, there must be some organization. Yeah, we will come to this now. Yes, there must be some kind of an organization. At that time, when that came out, I had the idea, no, it cannot be a passive diffusion. If it is a diffusion, you could see it, but this diffusion must happen actively. Because if it happens actively, then you have the chance to make it so exact. The proof that it must be active lies in the following. If you take a small organism or a large organism, you have the same proportions. That means the pattern is the same. If you had a merely passive diffusion, then of course bicoid, for example, could only travel so far because of its chemical physical properties and all that. So here in this, it could also travel only so far, right? Then you would have a distortion of the whole plan. That's not what happens, but the proportions are always kept up. So it must be an active process. And it did not take long that in Nature and Science and such magazines turned up the first reports that it is or it must be an active process.
Which year?
1999. In this book, it's on page 120. I have the quotation also. So, in Science, developing embryos may actively ship key signaling molecules from place to place, instead of relying on diffusion to carry the messages. And simple gradients can't explain the physical changes that accompany some crucial developmental events. Right? So this happened. Now we know that this is a very, very complicated process. First of all, we have these maternal pre-patterns. The maternal organism has to send 4 such different messenger RNAs at certain places. This has to happen at a certain time, in a certain place. Secondly, then you have the active production of the morphogenic substances. Thirdly, they are actively transported by transport proteins. Now, these proteins also have to be actively produced to be available at this time and in this place. Of course, their functional gene also had to be produced first by the activity of enzymes. Then we know that these proteins are absorbed by the nuclei actively, and then they are degraded actively again. And then we have the thing that we have 4 different such processes that happens all the time. How to coordinate this? if it happens at the same time at all these locations, which are dispersed among the, in the organism, there must be a coordination which is a non-local happening. It must be something which has to do with the whole, the organism as a whole, right? And therefore, one can say yes, orchestration, but an orchestration, and one talks also here of orchestration, an orchestration which has the effect that eventually, this is a quotation from Gregor from 2007, where he comes to the conclusion that the embryo, this embryo is not faced with noisy input signals, right? Something happening by chance, just by chemical physical processes and readout mechanisms. Rather, the system as a whole exerts precise control over absolute concentration differences and responds reliably to small concentration differences approaching the limit set by basic physical principles. So this happens flexibly in a quasi-liquid substance in space and time, and despite of the fact that it is flexible and it responds to disturbances, it is a goal-directed process with a precision which comes to the limit of our physical measuring instruments. This is an active deed of the organism as a whole. Now, because this activity has the effect of producing this Bauplan in time, one can say, well, the Bauplan itself is the goal, right? So actually, all these processes are arranged in such a way that the goal is arrived at, which means the processes do not produce the Bauplan. The Bauplan is, let's say, the prerequisite for this to happen. It has to be presupposed, but it's something which enacts itself in time. It is a causa finalis, final cause, but this final cause is a whole organization which has final temporal direction and arrives at its goal. That's why we have functioning organisms. It's rare that an organism has mistakes, in this. It's actually very, very rare. If you think about the complexity, take New York City, traffic. I mean, this is very simple, but just if one car stands like this, you have a traffic jam up to Spring Valley. So things like this don't happen in your organism. This is utterly flexible. So that's life. And life uses protein as instruments. For this reason, and with this I'll come to the next few, last few thoughts, because one sees, well, this must be a non-local process. Yes?
Would you say that the cause comes after the effect? It's like it's reversed.
The cause?
You have cause and effect, right?
Yes.
But the cause comes, in other words, the process is reversed. With a goal in mind,
Yes.
you have to do certain things.
That's right.
So you get to that.
You're right. But it is the same thing when you act, when you make a movement. You have a certain goal. This is in focus of your attention. And every movement that is functional or instrumental to arrive at that goal is a subsidiary element in order to arrive at that goal. Here, Polanyi with his thinking comes into place again. I think this is really great. Polanyi can think this way. This actually, the focus is on arriving the goal. That means the goal itself is the focus. And everything that happens is subsidiary in order to arrive at that goal. Right. Therefore, of course, you see the plan only after everything is done. That's why it's called a final cause, because it is fixed only after it's done. Just as when you make an artistic sculpture, after you make the whole process, then you can see it. Then it's also a final process, right? But because this happens really in a non-local process, and it must happen in a non-local process, because if you think that at the same time everything has to be coordinated, there is no time to have a signaling process of a material kind between what happens here and what happens here, if this must be coordinated in time, but in such a way that at the same time, at different places, there are actions which are instrumental for that goal. And this shows it must be a non-local process. Just in the same way as the two photons that we talked about today have no time, so to speak, to have an interchange on the material level, so here too, it's a non-material coordination. It does not function through external signaling processes, but it's a holistic process where a whole enacts itself in time and space. This is a natural consequence of a consequent thinking through of all what we know in biology. So, and because of this is really non-local, there appears a new now, sometimes, once in a while, in the literature, such things like, as here by Michael Levin from Boston, Tufts University, Boston, so near here actually. Morphogenetic fields in embryogenesis, regeneration, and cancer, non-local control of complex patterning in Biosystems 2012. Morphogenetic fields. Why fields? The term field is used here in analogy to physical force fields. Just as a magnet, for example, would create a pattern with its forces in iron powder that I would put on this table here, if I come from beneath, right, and then I can change, and you see how this pattern moves, etc., and arranges itself. So one can take this thought and can say, well, okay, so something which is holistic in kind, some kind of a force field must exist, which is responsible for all these changes to happen in these different localities. Holistic. Well, it is certainly justifiable to use this notion, but we have to see that there are decisive differences between a physical force field and a living force field, a morphogenetic field. Considerable differences. First of all, there is no source of no material source of the force in a magnet or in any physical force field. You always have some piece of matter from which these forces emanate. You have a magnet, you have a sender, right, for our smartphones. You have, if you take radioactive forces, they emanate from some substance, right? These forces do not, you cannot find any substance from which these forces emanate. And these forces that emanate from centers, they are also centric in their effect. They work externally on such a thing, right? These other forces are working internally. They permeate everything. And then they're also not rigid. A physical force field is always rigid. Of course, we know there are some fluctuations in, for example, Earth magnetism, but the Earth is probably also something else than just a physical element, right? But if you take a magnet, well, there's just a rigid force field, full stop. And also, the force field is changing in time. Take an embryo with these tremendous traveling of cells and the invagination, the gastrulation, all these processes. That's a sort of complicated process, which is in itself is a living process. So it must be a living force field, and it's organized as a whole, and etc. So we have remarkable differences, and for this reason, the term is not the best one, because it is also always associated with a physical force field, and then if we use it, then we tend to think too much in terms of physics, chemistry. Therefore, it's better to use another expression. That's why Rudolf Steiner talked of etheric body, because he explicitly wanted to have another term also to designate this higher class of forces which are acting in time and which are life per se. This organization is the life organization. That's the etheric body. So, but methodologically spoken, we can say that we are entitled to infer from all the facts we know in biology to make the hypothesis that such a force field must exist. In the very same way, we are entitled to say that there must be a force field when this iron beneath the table has the consequence that the iron powder is put into certain patterns. So we infer that. We do not see the force field, the etheric force field, but we don't see the physical force fields either. We always only see the effects in the physical, but we're not able, with our consciousness, to see the force fields. So the argument that, well, one cannot see these forces, and therefore it must be a hypothesis only, cannot be made, because if it would be made, one would also have to discard the hypothesis of physical force, right? Because in exactly the same way, in the biological realm, we can see effects of that. So we can infer that such a force field must exist. Of course, I know that many people get a bit nervous if you use terms like etheric or astral or something, because now they start to be very esoteric, and this has nothing to do with science. But you see, you need names. And when we talk about this, we have reasons to use such names, and also we can exactly say what we mean by that. So that is from the natural scientific point of view. Steiner does also talk from this point of view, but mostly from a spiritual scientific point of view. So the question would be, how could one have an access to these force fields in a spiritual way? Which will be the topic of the last lecture on Saturday. But to come to a close, the question whether really life can be explained in chemical or genetic terms, no, it cannot. It's not physical and chemical or biochemical forces which are the agency of life. But it is, as we said, this organized system of self-organizing spatial and temporal laws which enact themselves. And the interesting thing is that Steiner also says that the etheric body is like the architect of the physical body. And this is interesting because if you think, what do regulatory enzymes really do? What does an enzyme do? An enzyme doesn't regulate, you know. It's not a regulatory entity, but what an enzyme does, it allows the effect, it allows the self-organization of something else than it is. For example, if you have a chemical, a catalyzer, if you have a substance A which goes to B, and you have an enzyme here, the enzyme does not determine the lawfulness of A or B, but it also only facilitates the self-organization of this process. The same thing happens in the organism. The enzymes facilitate the self-organization of these conformational the physical form principles in time. That's what they do. And therefore, it is correct to say they're like the architect, which the architect allows, or of course he does it actively, to form the material according to these laws. But these laws are different laws than the architect. And so the laws which are allowed to act are other laws than the laws of these enzymes. So we see the actual agency or causality that is active here is this unity of this temporal and spatial organization. So definitely there is no chemical explanation of life. But I would just like to add as a last sentence that you find in the present literature interesting new avenues. For example, the book by Dennis Noble of 2008, The Music of Life, Biology Beyond Genes, right? Musicality. That's where one comes to it. There is a musicality, not the genes, but it's beyond genes. Or Brian Henning and Adam Scarfi, 2013, Beyond Mechanism, Putting Life Back into Biology. Yes, that's what you have to do, and that's why we talked about this tonight. Thank you.